All right, next lecture. This is also being broadcast as an SDL. The next thing we're going to talk about is autosomal inheritance, which there's a 1 and a 2, so it's actually a 2-hour lecture. I'm just going to do the first part now, and we'll do the second part live on Wednesday morning. So there's one PowerPoint that covers both of the lectures, and so I'll stop about approximately the halfway point through this lecture. And it's just continuing on with our topics in genetics. And by the way, I realize I'm not doing this with a live audience, so and therefore you don't have the chance to come up after class and ask me questions. Remember, you can always email me questions, and of course I'll be there in class on Wednesday as well. So don't feel like you can't ask me questions. Hopefully I'll be able to answer them, any questions that you have. If they're quick questions, just a clarification or something like that, usually I can hammer that out real quickly on an email. If it's something that requires an explanation, I usually like to do it in person, either at the end of class or something like that, or in my office, just because I want to make sure the choice of words that I use, that you really understood my explanation. Okay, so before we actually start looking at the inheritance, we need to define some terms that we're going to use in the section on genetics. So these will be terms that not only I will use, but Dr. Gardner may use as well. And so I just want to go over what I mean by these terms. The first is gene. And the definition we're going to use is a basic unit of inheritance. Gene is probably the one definition that we're going to use in here that's not really super well defined, because what is a gene has changed over time. And so now that we understand in eukaryotes how genes work, and enhancer regions where they can be located, could potentially even be on different chromosomes, it becomes very hard to define a gene, because it used to be a gene was what ends up in the mRNA, and then the introns in the promoter region, and that basically made up the gene, so all those things. But now that we realize that it's a little more complicated than that, and that you can also get multiple protein products from a single gene, things become skewed a little bit. So the term is probably the most frequently used term in the whole section on genetics, not just by me, but by textbooks as well. But it's the true definition of a gene is a little bit controversial, shall we say. Locus is, or loci, if it's multiple, is the physical location of a gene on a chromosome. So, and for some diseases, we will talk about the actual physical location. Allele, alternative forms of a gene at a given locus. So for allele, normally in this class, we're really going to be talking about a normal and a disease allele. That's the extent we're going to get into. But for those of you who've had genetics before or want a little bit deeper understanding, there certainly can be multiple different normal alleles. Okay, and they're going to be different at the sequence level and the amino acid level, perhaps, but they don't perform differently in a measurable way. Therefore, they don't provide any genetic advantage or disadvantage, so they're genetically neutral. So you can have multiple normal alleles. You could also have multiple possible alleles that cause disease as well, and some of those may be more severe than others. Okay, so alleles are just different forms of a gene. Genotype and phenotype. So the genotype is the genes that you actually have. Okay, and for most of the genes we're going to talk about, you have 2 copies of each gene. And then we have the phenotype, which is the observed expression of the gene. And these two don't always line up. So the phenotype is what you see. The genotype is what's actually there. So the genotype plays a role in the phenotype, but there's other things going on as well. And some of these affect the inheritance pattern, whether things are dominant or recessive, as we're going to see. You can also see these terms homozygous or the homozygote and the heterozygous or heterozygote. Homozygous, you have two identical alleles at a given locus, where the heterozygote has two different alleles. And once again, most of the time we're talking about, when we go through the section on medical genetics, we're talking about a normal allele and a disease allele. So you can have one copy of each and be a heterozygote, or you could be homozygous for the mutant gene or homozygous for the normal gene. Okay? And then the dominant and recessive, which we're going to start looking at today in terms of the inheritance patterns. Dominant means a condition seen in both the homozygote and the heterozygote. That means only one copy of the allele we're talking about, and usually we're talking about, in this class, we're going to be talking about mutant alleles, is necessary for phenotypic expression. Now, you can use these terms dominant and recessive for things that have nothing to do with disease as well, which is certainly, if you go back to Mendel's original experiments, he was looking at. He wasn't really talking about a disease in particular, but in this class, we're really only going to be talking about diseases when we're talking. So we're really just going to have normal or mutant. And so whether a disease is dominant or recessive depends on whether you need one copy. If you need just one copy, then it's a dominant condition. And if you need two copies of the mutant allele, then it's a recessive condition. So it is really important, and I put this little star here, to realize dominant. When we say dominant and recessive, we're referring to the phenotypic or the phenotypic expression. Okay, not the expression of the gene itself. Okay, let me explain what I mean by that. In most conditions that we're going to talk about, in most of the diseases we're going to talk about, as well as most of the genes in your body, you have two copies of those genes, and you express both of them. There certainly are exceptions out there, and there's certainly, and when we talk about X-linked, things are going to be a little bit different. And there's for some exceptions you'll see with some diseases. But for the most part, unless you're told differently, you can always assume that you're seeing expression from both genes. Whether the condition is dominant or recessive is aside from the fact that you're getting expression from both genes. Both the mutant allele, if you're talking about a heterozygote, both the mutant allele and the normal allele. Or if you're talking about a homozygote, both normal alleles, or somebody who has an autosomal recessive condition, both mutant alleles, they're always both being expressed with a handful of exceptions. Autosome, these are the non-sex chromosomes. So your sex chromosomes are your X and your Y chromosome. Everything else is an autosome. And there's 22 autosomes in humans, and they're numbered 1 through 22. And you have 2 copies of each of those autosomes. Diploid refers to the fact that you have 2 copies of each chromosome in a particular cell, so we call that diploid. You could also have cells that are haploid. You could have organisms that are haploid, but you're actually a diploid organism. That means you only have a single copy of each chromosome, and really the only place you see that is going to be in your gametes. So sperm or egg cells are going to be haploid. That's because when they come together, they're going to make a diploid. And all the other cells besides gametes are somatic cells, and all of your somatic cells are diploid. Okay? So you'll see that term somatic used in some other places as well. So there's 3 major types of genetic disease. And the first is your single gene disorders. And really that's what we're going to spend most of our time talking about in the genetics section. Dr. Gardner is going to get to some chromosomal abnormalities and multifactorial traits in a couple of lectures. But most of the time we're talking about single gene disorders, monogenic disorders, sometimes also called Mendelian because they follow Mendelian genetics. These are disorders produced by the effect of a single gene, or you can say a single gene pair. And they're usually transmitted in simple patterns. And we're going to look at those patterns: autosomal dominant, autosomal recessive, or X-linked. And you can actually look at pedigrees and be able to distinguish these unique patterns. You also have, in terms of genetic disease, you also have your chromosomal abnormalities. Down syndrome may be one you've heard about, but in these cases, you have a deviation from the normal chromosome number or structure. There's also some structural differences. So, in some diseases where you're missing a piece of a chromosome, or you've got an extra piece of a chromosome. Dr. Gardner will talk to you about some of these. And then you have multifactorial traits. And these combine the effect of multiple different genes and also non-genetic influences, environmental influences, for instance. And some of your biggest diseases are multifactorial. Things like heart disease, things like type 2 diabetes. They can be multifactorial, means the environment can also affect it, and there's more than one gene. Cancer is another one. So the multifactorial traits, Dr. Gardner will talk to you a little bit about, but from a genetics perspective, it becomes very complicated to talk about. It's very easy to talk about single gene disorders because we've narrowed it down. We know the gene. Very often we know the exact defect, and we can rehearse or recite everything from the molecular level. With multifactorial traits, there's all different influences. Things like heart disease, you can get a genetic analysis done and get your estimated risk for heart disease, and each of these different genes and alleles that they're looking at may have a 1% or a 2% influence on your total risk. So there's just many genes involved, and certainly environmental factors as well. So one of the things we're going to be looking at now is we're going to look at some pedigrees. One of the things I want you to be able to do is interpret a pedigree. I want you to be able to go through the basics of reading a pedigree, which we're going to go through right now. And also, I want you to be able to tell me, what type of pedigree are we looking at? Is it an autosomal recessive? Is it an autosomal dominant? Is it an X-linked? Is it a mitochondrial? And we'll go through those. So some basics about reading the pedigrees, okay? The squares are males, the circles are females. In general, in a pedigree where you see something shaded in, that means you have somebody affected by whatever disease that pedigree is trying to represent. Okay, so in here we have a couple places where you see somebody who's affected. When you see that horizontal line, you're talking about mating. So here is a man and a woman, and they have 4 children. Okay, so you can see there are 4 children there. And then their children go on, at least some of them, and have children. And we're going down through generations. Okay, so each step is a generation as well. Now, that's really the basics of reading a pedigree. If you know what I told you right now, you know probably 95% of the pedigrees that you're gonna come across. Now, there are a few things that you may come across that are listed here, and we'll go over some of them real briefly, because some of them we will see in this class. You may see unknown sex, where you have the diamond shape there. Usually when we say unknown sex, we're talking about a fetus, and that's why we don't know the sex yet. So we're talking about doing some kind of genetic testing. We haven't determined the sex yet. And so, at this point we don't know. Now, there are a couple different ways where they can show carriers in a pedigree, but one thing I want you to understand is most of the time, unless they're trying to illustrate something in particular about the carriers, most of the time they don't show you the carriers. So when we're talking about an autosomal recessive disease, for instance, there's going to be carriers in the pedigree. And one of the things I'm going to ask you to be able to do is go back and figure out who is a carrier, or whether a certain person was a carrier or not. And so it's important for you to recognize in those, most of the time we don't see the carriers designated, okay? Sometimes you will, and if you do, you'll see one of these two things, and I think there's probably, in the section on genetics before we're done, you'll probably see both of these represented, either a half shade or a circle inside. I've seen the circle inside used also for carriers for autosomal recessive, or where we had one example that we're going to see later on is where we have incomplete penetrance in something that's supposed to be autosomal dominant, and they indicated the carrier in that case with this little dot. So you will see that. Some other things you may see, you can have twins in there, and don't let that confuse you. There you have your non-identical twins, and these are identical twins. This is the more conventional version on the right for showing identical twins rather than this triangle. They just put the line right there. That's the more conventional way, but you could see either one. One thing you may see also in a pedigree is a double line. Double line indicates incestuous or consanguineous matings. These are matings between closely related individuals. In this case, we have a mating between a brother and sister. More commonly, you'll see in pedigrees, you'll see matings between, consanguineous matings between cousins, for example. And we'll talk a little bit about what the consequences of consanguinity are a little bit later on. I can let you know right now that it's not going to be a good thing genetically. Okay, so the first pedigree we're going to look at is our autosomal dominant pedigree. So one of the things I want you to be able to do is I want you to be able to look at a pedigree and tell me whether it's autosomal dominant, recessive, X-linked. So eventually we'll get to all of those, mitochondrial. And so one of the things I want you to do is, because we're not going to cover all of them until we get through Thursday, at the end I want you to be able to differentiate between them. So make sure you can. So make sure at the end, after we've seen all of them, you can look at this and say, okay, that's autosomal dominant. Why is it autosomal dominant? Well, because of these reasons. So first of all, with autosomal dominant disease, we only need one mutant allele for the disease to show up as a phenotype. Okay, that's what we mean by autosomal dominant. If you've got one mutant allele, you have the disease. So, and in most cases, most people with an autosomal dominant disease only have a single copy of the mutant allele, most of the time. That means an affected person has at least one affected parent. So you have this vertical transmission. You don't see generation skipping. Okay, you can follow right down the pedigree from person to person from person. You have a parent with a kid, and then they have a kid, then they have a kid, and you can follow it down vertically. Okay, either sex can be affected, so there's no dominance of males versus females. Okay, that's going to be true for both autosomal type of pedigrees. And also for both type of autosomal pedigrees, you can get male-to-male transmission. So these two factors are going to differentiate from some other pedigrees we're going to see later on. Okay? So our autosomal dominant, and one thing to remember is, and I didn't explicitly state it here, but remember every disease has to start somewhere. Okay, there has to be the first person who has the mutation. In this case, in this pedigree, it's right here. It's possible to have a pedigree that goes further even back and you don't have incidence of the disease. So this person could be the very first person. And you can write this in, they call that the proband, P-R-O-B-A-N-D, because I believe Dr. Gardner will use that term a little bit later on. So that's the first person to show up in a pedigree. But you can safely assume that new mutations are very rare, so you're not going to see a whole bunch of new mutations in a pedigree. Usually a new mutation is only going to occur once. And one thing also about the autosomal dominant pedigrees is they often involve non-catalytic proteins. Proteins that are not enzymes. Or another way of saying it is structural proteins. Okay? When you mutate collagen, for example, that would be something that would end up showing up as a dominant disease. And we'll explain why that is in 1 second. Here's some very famous, very high yield disease examples. Pretty much if I mention a disease in the genetics section, it's a disease that you absolutely need to know for board exams. There's only one real rare disease that I may mention, and I'll point it out at that time. Everything else, if I'm going through it and you're like, well, he told us some things about this disease. I wonder if we need to know them. Just know them. It's going to make your life easier. I may not be able to test you on every detail, but you're going to need to know these diseases inside and out. Okay. Right now, we're focusing more on the genetics, but for some of these diseases, I'm going to give you a little bit of the pathology and things like that. And you should just go ahead and learn them because you're going to have to know these ones inside and out. Familial hypercholesterolemia, very famous, very prominent for a single gene disorder as well. About 1 in 500 people have this condition, and it's a defect in the LDL receptor. And why do you get? Why is that autosomal dominant? I'll explain in a second. So things like receptors or other things involved in signal transduction, those tend to be dominant. When you get a new, so that means there's one copy is enough to cause the disease. That means that the normal copy is not enough to cover for the mutant copy. So you might think I would guess, just not knowing anything else, for the receptor, who cares if you've got a mutant copy? Just make more of it from the normal copy, but it doesn't work that way in the receptors and these signal transduction pathways. So those types of mutations tend to be autosomal dominant. Huntington disease, there's a disease we've already talked about. And that is also inherited in an autosomal dominant fashion, as well as myotonic dystrophy. Both of those were trinucleotide repeat expansions, and both of those, you actually had a gain of toxic function, which I'm going to get to in a second. So the dominant allele, in this case, the disease allele, causes problems. Okay, it's not just that it's non-functional, it also interferes with things. And then, therefore, if you got one thing interfering with things, then it causes a problem. Okay? NF type 1, neurofibromatosis type 1, we're going to talk about that disease today, as well as we're also going to talk about Marfan syndrome. So I don't know if you've seen these. If you haven't, we're going to talk about those ones today. Osteogenesis imperfecta 1 and 2, we definitely have talked about those. Those are autosomal dominant. There are some versions of osteogenesis imperfecta which are recessive. And here's a good question for me, just to throw out, just for an understanding type of thing. It's not something I would necessarily test you on. But how can we have osteogenesis imperfecta that would be autosomal recessive? Okay. So osteogenesis imperfecta is a good example of a disease where you have a structural protein. If you have a defect in 50% of the structure, well, your whole structure is defective. If I build a house and half the wood is defective, well, that whole house is defective, right? Whereas, let's suppose I build that house and I have 2 hammers. One of them doesn't work right, but the other one does. I can still build the whole house, okay? And that would be like an autosomal recessive condition, where the catalytic thing in the hammer, in this case, could actually build a whole house and the structure would be okay. So the structural proteins, because they, having 50% of the structure being defective interferes with the entire structure, those tend to be dominant diseases. So anytime you see, just without knowing anything else, if you see something that happens to be a structural protein, you can guess if there's a disease involved with it, if there's a known mutated allele, it's probably autosomal dominant, if it's the gene's located on an autosome. Certainly in some cases the gene could be located on the X chromosome, and that changes things a little bit. One of the things I want you to be able to do with the autosomal dominant and also the autosomal recessive diseases is calculate the recurrence risk. Or sometimes you also see it referred to as the occurrence risk if it's the very first child that somebody's having. But very often they just use the term recurrence, whether the couple has had a child or not. Okay, so it's the probability, basically what we're trying to calculate is the probability that offspring of a couple will have a genetic disease. So these are just going to be our little Punnett squares, which you've probably all done before, right? One thing to remember, okay, important thing to remember, each reproductive event is statistically independent of all previous events, okay? So if I calculate the risk here, you know, okay, well there's a 50% risk, and they've had 5 children, and every one of them already have the disease, or every one of them don't have the disease. How does that change, if they're having another child, how does that change the percentage of chance they have of having the disease? It doesn't. That's what statistically independent means. You know, if you have a 1 in 4 chance of having a child with a particular disease, okay, and you had 6 children in a row and they all have the disease, okay, you might think, well, the chances of having another one have to be much, much lower. No, it's still 1 in 4. Okay, so that's what we mean by statistically independent. So when you're trying to calculate these things, you need to just focus on what the genotype is and do these Punnett squares. It doesn't matter how many children they've already had and how many of those have been affected or unaffected. Okay? So we're doing an autosomal dominant pedigree here, or not pedigree, but Punnett square, if you will. So we're doing a cross. Now, by convention, the uppercase is the dominant allele and the lowercase is the recessive allele. This doesn't tell you whether or not we're talking about a dominant or recessive disease. That's just by convention the way they do it. Uppercase means dominant, lowercase means recessive. Okay? So you have to figure out for yourself when you do these crosses, well, am I talking about a dominant disease when I go to draw these out? I'm talking about a person who has a dominant disease, and the majority of the time somebody who has a dominant genetic disease only has a single copy of this mutant allele. They would be, in this case if we're assigning the allele A, big A, little a. And then somebody who's normal, if they're normal, then if we're talking about a dominant disease, then both of their genes have to be normal, then it would be little a, little a. Okay? And actually, this would be your most common cross with autosomal dominant, is you have a person with the disease who just has a single copy of the gene, which is the majority of these people who have whatever disease you're talking about. It's quite rare to get somebody who gets 2 copies of an autosomal dominant disease gene. And then with the normal individual. So we're crossing these 2. Okay, so that's your most common cross. Now, what's the chance of them having an affected offspring? So you just make this little square, okay, if you haven't done this before. You break up the genotype into lines, okay? And so you just draw a blank square. So, and then you have little a, that's one person. And over here you have big A, little a, that's another person. And then you just follow along. Let's see, A, little a, and big A. Okay, that's one. Now down here. Big A and little a. That's another one. Over here. Little a, little a. There's another one. Little a, little a. There's another one. Hopefully you can see that. If you struggle, this should be something that's pretty simple. Once again, it's one of those things I'm not going to belabor it any more than what I just explained. If that didn't make sense to you or you're struggling with that, come see me because we can iron it out super fast trying to do it in person. Okay? So in this case, if we talk about somebody who has an autosomal dominant disease and they just have 1 copy of the gene, which is most of them, versus a normal person, they have about a 50% chance of, or they do have a 50% chance of having an affected offspring. Okay? Now, when we're doing these crosses, we're assuming that whatever the disease is not going to kill the fetus in utero, so that they would never be born. So these have to be diseases which you can at least survive to term with. And there, I've given you a question, and this is something you should be able to do. Let's take, for example, we have somebody, two people who have this autosomal dominant disease. And in both cases, they have one copy of the particular gene. What's the chances that they would have an affected offspring? So go ahead and do that on your own, and I can give you the answers later. And what happens if you have a normal individual and they happen to cross with somebody who's got two copies of an autosomal dominant disease gene? What would end up happening in that case? Try to do those yourself. They're pretty simple. Another way I could potentially ask a question is I could give you a pedigree, and one of the things you would need to do is look at that pedigree and say, okay, see the disease in every generation, males and females equally. Okay, I can figure out that's an autosomal dominant disease. In all likelihood, that's an autosomal dominant disease. And then I could say something like this. What is the genotype of the woman in the uppermost row? Okay, I figured out that's autosomal dominant. Now he's asking about this woman. Now she's affected. Talking about an autosomal dominant disease, so that eliminates this first choice. She can't be little a, right? Because she's showing the disease. So that automatically means it has to be one of these two. Okay, now we look at her children. Well, she's got children that are both affected and unaffected. Okay, so that means she was able to pass on either a mutant dominant allele or the normal recessive allele. Okay, so that means we can actually say definitively that that person is big A, little a. This is somebody who has an autosomal dominant disease who has a single copy of the gene in that case. Now in this case, you'll notice she has 6 children, and they happen to come out 3 affected and 3 non-affected, so 50-50. Remember, you know, each thing is, you could certainly have 4 and 2 or 5 and 1 or possibly even 6 and 0 with a dominant disease, just by chance, okay? So that's something I can ask you. Another thing that I can ask with these pedigrees also is I may give you a pedigree and say, and give you a list of diseases and say, which of these diseases could fit that pedigree, okay? So in order to be able to answer that question, you first have to be able to figure out, okay, this is autosomal dominant, and then look at the diseases and know which ones are autosomal dominant. Okay, so that means earlier on in the slide, I would like you to know that these diseases are autosomal dominant. These are all big, high-yield diseases. Some of them we've already seen, some of them we're going to see today. The only one you may not have seen yet is familial hypercholesterolemia that we're not going to talk about today, but you'll certainly see that in the section on fat metabolism. Actually, Dr. Traore's probably already talked to you about that, so you probably have seen that. Okay, so within the autosomal dominant diseases, we can actually break them into 2 general types. You have your loss of function mutations, and then conversely, we can have our gain of function mutations. So within the loss of function mutations, we can have a couple different examples. And I think the easiest one to understand is when you have a defect in a structural protein. And a very good example is osteogenesis imperfecta, the type 1 and the type 2. Both of those, one's more severe than the other, but in both of those you have collagen mutations, okay? It affects the structure of collagen. Once you affect the structure, you affect the entire structure. Okay? That's why it's dominant, okay? And I think those are the easiest ones to get in your head. The other thing that can cause, the other place we can have a loss of function mutation is in regulatory proteins. I like to think of these as proteins involved in signal transduction. These could be receptors or something else involved in signal transduction. Once again, these are a loss of function mutation. So one of the mutant gene in this case lost its function. It's not working correctly. LDL receptor deficiency in familial hypercholesterolemia, very good example of that. If you only have one normal copy of the LDL receptor, it will be able to function normally, but you will still have the symptoms of the disease because you don't have enough of it. And with these things involved, receptors and other things involved in signal transduction, the amount that's present is quite critical. It is regulated to very tightly. And if one of the genes is defective, and 50% of the protein that you're making for that pathway is not working correctly, it affects the whole pathway. Okay? It still works, but it's not working as good as it should, and therefore you end up with symptoms of a disease. So this is something we would call haploinsufficiency, where one copy of a normal gene is not enough to provide normal function. So you can contrast, this is a very good thing to contrast with our autosomal recessive diseases, because in those diseases you have haplosufficiency, where one normal copy is enough to do the job. We'll talk about those a little bit later. So we have our loss of function mutations, and then we can also have our gain of function mutations. Okay. You'll also see sometimes people call this dominant negative, but I call it gain of function mutations. Gain of function mutations, unfortunately, it sounds like that's a good thing, right? Hey, we gained a function. We should be happy. We're doing something new. So I like to call it gain of toxic function, to remember it's really something bad. So you're gaining a toxic function. And this is when you have a mutation that endows some normal protein, or in some cases it's actually the RNA. Whoops. In some cases it's actually the RNA with toxic properties. Huntington disease, a very classical example of a gain of function mutation. Okay, where something now has toxic properties. So if something's toxic, even if only 50% of it's there, if it's toxic, it's going to affect the whole cell, right? So that's why that would be dominant. Huntington disease, you have a defective Huntington protein. It precipitates out of the cell or within the cell, and that precipitation of this abnormal protein ends up killing the cell, and it destroys neurons. Okay, so in that case, you only needed one copy in order to cause some type of effect. Okay, so we're going to look at a couple of autosomal dominant diseases. A couple that we mentioned earlier that we haven't explained yet, or there isn't a convenient place in other places in biochemistry to talk about. I think I've talked to you about all of the others, with the exception of familial hypercholesterolemia, which I believe Dr. Traore has talked to you about. The ones we haven't talked about, we're going to talk about right now. So, neurofibromatosis type 1, NF type 1, it's much easier to say NF type 1. Very famous, very well-known genetic disease, one you will absolutely be required to know for board exams, NF type 1. It is autosomal dominant. Okay, the gene defect in this case is a mutation in a regulatory protein. It's actually called neurofibromin, and the gene is called the NF1 gene. Now, very typically, you won't always see this, but very typically, when we're talking about the protein, they won't use italicized, and when they're talking about the gene, they use the italicized version. And in this case, when we're talking about NF type 1, if you don't see it italicized, they're talking about, when they see this NF1, here we're talking about the gene, here we're talking about the disease. So that's just an FYI, in case you're confused when you're going through why it looks that way. So this is a protein that would normally be involved in controlling the cell cycle. It's also something that would be considered a tumor suppressor gene. Okay? So, you can start to imagine, okay, well, if I have a defect in the tumor suppressor gene, what's actually going to end up happening? The incidence of this is 1 in 3,000. Now, I'm giving you incidence rates for some of these diseases, not because I expect you to try to memorize the incidences, okay? You're welcome to do that. It's certainly not going to hurt you, and I think it's an interesting thing to know, and it can help to a certain extent to know whether things are common or not. But that's the big thing. I want you to realize when you see a number, whether we're talking about a rare genetic disease or a common genetic disease. And 1 in 3,000 is, for a genetic disease, is extremely common, okay? I usually put the cutoff somewhere around 1 in 10,000. If you see it less frequently than that, you're talking about something that's starting to get pretty rare. If you see it more frequently than 1 in 10,000, then you're talking about something that would be a common genetic disease. Although it's still, 1 in 3,000, you don't see it in a lot of people, and you don't see it necessarily every day. From a genetic perspective, for a genetic disease, that's actually extremely common. The NF1 gene is very large. It occupies a section about 350 kb, so it's one of the largest genes. And the bigger, the more space a gene occupies on the chromosome, the greater the chance that it's going to become mutated. And so NF1 has a very high incidence of new mutations. About 50% of the cases are actually due to new mutations in the NF1 gene. Okay? Now, obviously, if I'm doing a pedigree, I can't have a whole bunch of new mutations. So don't let that screw you up when you're looking at a pedigree. So if I give you a pedigree and I'm talking about NF type 1, it's going to look like your traditional autosomal dominant pedigree. Okay? So some of the clinical picture about NF type 1, some of the symptoms that you will see. Okay, cafe au lait spots, that means coffee and milk spots. So that's what you're seeing here on this individual. Okay, very characteristic of NF type 1. Here's the big thing for NF type 1. You have these multiple neurofibromas, and what these are benign peripheral nerve tumors. Okay, now I'm giving you some of the most extreme cases that I could find pictures of, but if both the people in this photograph and this person both have NF type 1, and you're not seeing tons of neurofibromas here, are you? So this is a disease that I'm going to talk about again in the complications of the pedigrees, where you can have a real wide variety of expression of symptoms of this particular disease. And I've given you, once again, most people with NF type 1 aren't this severe. I've given you some of the most severe versions. Now, these neurofibromas, and sometimes you can have somebody who have a single neurofibroma that just gets absolutely enormous. So if you could type this into, I didn't put one of those on here, but you can type into Google Images and see that. You'll just have a single one, but it'll be absolutely enormous. The danger here, besides the aesthetic effect and the things that come along with that, are that these can potentially develop into something worse, and these people can develop malignant peripheral nerve sheath tumors. So you can actually develop eventually into cancer. And in some cases, some of these neurofibromas, even if they're not cancerous, can get so big that they end up interfering with daily functions as well. Axillary freckling is something characteristic of NF type 1, so you got freckles on the armpits there. And you can see this person doesn't have any neurofibromas in that section where you can see, so they're not as severely affected. Another big symptom that's very characteristic of only NF type 1 is Lisch nodules. Okay, these are benign hamartomas. So a hamartoma is something that, it looks like a neoplasm, it looks like some type of tumor, but it's still retaining some of the properties of the original tissue which it's derived from, and it's going to be, in most cases, benign as well. So you get these, but you can almost think of these as being very similar to a neurofibroma, and it's just on the iris of the eye. This is something very characteristic of neurofibromatosis type 1. It's one of those things that you may see on a board exam. I would certainly, this would be the type of thing that the COMLEX would like to show just a picture of, okay, and not tell you that it's called a Lisch nodule, but have you recognize, oh, what's going on there? That's the iris. Looks like you got little tumors, and recognize that it is a Lisch nodule, and be able to tell them that it's NF type 1. Okay. So we're actually going to revisit NF type 1 in a little bit later lecture. And we're also going to revisit Marfan syndrome as well in a little bit later lecture, because they also are prototypical diseases to highlight something else that I want to talk about. So Marfan syndrome is the next disease. Also autosomal dominant. We're still talking about our autosomal dominant disease. With Marfan syndrome, the most common defect, but by far, most of the cases of Marfan syndrome are by a mutation in the FBN1 gene that encodes fibrillin 1. So you've actually got multiple different possible, fibrillin genes. Fibrillin, the protein, is a major component of the extracellular matrix. So it's a key component of connective tissue. In this version of fibrillin, you see the major expression of this in the aorta, the periosteum. The periosteum, and maybe you guys already know what the periosteum is. It's not a term that I use every day, so I wrote it down for myself if you already knew what it was. But if you didn't, it's a dense layer of vascular tissue covering the surface of bones. Almost looks like a skin on the surface of bones. It's dense, but it's very thin as well. And the suspensory ligament of the lens. These have major expression of fibrillin 1, and therefore you might be able to guess, we're talking about an autosomal dominant disease. This could be, those might be tissues that would be affected. Okay, this is also one where we're talking about something that's a non-catalytic protein, a structural protein, therefore we have an autosomal dominant disease. Okay, the last example of the NF type 1 would be something where you have a loss of function, where you have a regulatory protein where it loses its function. So something involved in signal transduction. So some of the symptoms you'll see in Marfan syndrome because of the tissues which are affected. So you're going to see effects on the bones, see skeletal abnormalities. And you get a wide range of symptoms. So in some cases I picked some of the most severe things I could find. So here's a patient with Marfan syndrome, and you can actually see some of the other symptoms that we're going to get at later on, or in the very next slide with Marfan syndrome. But you can really see the scoliosis in this patient from the skeletal abnormality. Typically, people with Marfan syndrome have long, thin limbs. They tend to be very tall. They have very long fingers and toes. Okay, it's very characteristic of persons with Marfan syndrome. Also, they have hypermobile joints. So this is something that in some cases can, in certain ways, it can resemble particular types of Ehlers-Danlos syndrome. So you have to be able to differentiate between them. You also see ocular abnormalities. Remember, it's involved in the suspensory ligament of the lens, so you can get a detached lens and also myopia. Very characteristic of Marfan syndrome. And the big deal with Marfan syndrome is the cardiovascular disease that you can see. Okay, and so you can get an aortic aneurysm, which eventually could become life-threatening in the case of someone with Marfan syndrome. So here's a question, and I used to get asked this very frequently, so I decided to include a slide here and first of all, pose the question for you guys. What happens if a person inherits 2 copies of a defective autosomal dominant gene? It's always something that pops into people's heads. And it's also a good point to think about also when you're talking about the terms dominant and recessive. Sometimes the lines start to get a little bit blurred. Okay, and we're really going to see that when we talk about the thalassemias, for instance. So we consider them autosomal recessive, but even in someone with 1 mutant copy could still show some symptoms. So why is that? Well, we'll talk about that in the next lecture. But what happens if somebody inherits 2 copies of a defective autosomal dominant gene? In most cases, the disease phenotype is going to be more severe. There are exceptions. There are autosomal dominant diseases where whether you have 1 or 2 copies of a mutant gene, it still manifests the same. But those are the exception more than the rule. Most of the time, if you get 2 defective copies, you have a much more severe disease. Okay? Familial hypercholesterolemia is not one that I have on this slide. That one's common enough that you will occasionally have a patient, instead of 1 in 500, it happens to be about 1 in a million, where they will have 2 copies of that defective LDL receptor, and they have a much more severe course of disease. Okay. Another example where you could see this is achondroplasia. So achondroplasia, I don't know if I said it here, no. It's the most common form of this short-limbed type of dwarfism. Okay? It's due to a very specific point mutation, very, very specific mutation, and it is autosomal dominant. So you have a very specific point mutation in a gene called FGFR3. This is a fibroblast growth factor receptor. So that's what the FGFR stand for. Okay? It's a very, very specific mutation that causes achondroplasia. And this is an autosomal dominant disease. That means you only need 1 copy of that particular gene to show the symptoms of the disease. And I'm sure I've got a picture right here of somebody with achondroplasia. People with achondroplasia, they have a short-limbed dwarfism, so their torso is approximately average size, but their limbs are much shorter. Their head is of normal size. This is something that I think you probably all recognize. There's some very famous people who have this type of dwarfism, and because it's so common, it's one of those things, kind of like Down syndrome, where once you see it and you kind of recognize it, you're like, oh yeah, I can see what that person has. I don't really watch Game of Thrones, but there's an actor on Game of Thrones who has achondroplasia. There's some other things. If you ever watch Little People, Big World, the mother and Zach, the son, both have achondroplasia. So there's a good question, genetics question for you. In that family, what was the chance of them having a child with achondroplasia? So calculate that. But in people who have achondroplasia, there are health things associated with it, such that the average lifespan of an individual with achondroplasia is reduced by about 10 years. They can certainly live as long as a normal person, but overall, when you average it out, it's reduced by about 10 years because of some of the complications. But that's compatible with a relatively normal life. However, if you get 2 people with if you get 2 people who have achondroplasia and they have a child who inherits 2 copies of that mutated gene, that is going to be lethal, either in utero or in infancy. Okay, so in that case, the effect of having 2 copies was much more severe. And this is a realistic probability. You will see, because dwarfs come together in support groups and meet one another, so it's very common for a dwarf to want to marry another dwarf. But if they both have the same type of dwarfism and they both have this achondroplasia, they have to be aware of the risks if they're going to have children. So there's a good exercise for you. If you have two people with achondroplasia, what's the chance of them having somebody, a child who has gets 2 copies of the abnormal gene, or a child that has achondroplasia where they just have 1 copy of the mutant gene, or could they have a child who could be of normal height? And that's a cross that you guys can do. I think that's actually one of the exercises I gave you. Okay, so I'm going to stop here, and next time we'll pick up with the autosomal recessive diseases.